Okay, continuing on here on the immune system. Earlier we were comparing specific versus non-specific immunity. I just wanted to reiterate that really quick. And we're talking about why you should not be worried even if you do contract coronavirus in this section on why they want to hide your immune system from you. In the non-specific immunity, the basis of non-specific cellular mechanism defense is centered on the ability of certain white blood cells to ingest and digest microorganisms. There are two types, white cells, granulocytes, like I said, and macrophages. Okay? And so they can act, like this chart right here, macrophage, mast cells, basophils, neutrophils, eosinophils, platelets, they can act alone, but sometimes they may require more, and that's when the specific immunity gets involved, which is T and B cells. B cells and T lymphocytes. Okay. Now, I go over to page 175. Okay. On viruses. Okay. So, once the virus penetrates the cell, the former is protected from any antibodies that may be circulating around the blood. So remember I said that under specific immunity, T cells are intracellular and the B cells are outside the cell. So once the virus gets inside the cell, it's immune to the B cells, which are your antibodies. Here we have a nice chart, or a nice schematic of host cell, and then here's a virus with surface antigens. Okay, immune response is possible. We go immune response. Now, once the virus goes inside the cell, like we have here, immune response is not possible. Virus replication is inside the infected cell. Then we go down. Once it's inside the infected cell, these are the virus particles on the outside of the cell. Then an immune response is possible. Virus proteins incorporated in host membrane. And then we have an immune response possible, where we have immune response, assembled virus released from the infected cell, and then you have immune response possible once again. Viruses are intracellular parasites. Okay, that right there, we can read it. That usually invade epithelial surfaces and then enter the blood, viremic phase, before reaching their target organ. Specific immune responses are activated only by the surface antigen, a free virus, or by viral proteins exposed on the surface of viral infected cells. Response to viral antigens is almost exclusively T cell dependent. That is, even B cell activation requires T cell help. Individuals with T cell dysfunction are especially susceptible to viral infections. Viruses released into the bloodstream can be attacked. Now, viruses released into the bloodstream can be attacked directly by antibodies, such as IgG, IgM, and neutralized. Once a virus reaches a target organ, it is subject to specific effects of antibody, complement, and cell-mediated responses, as well as non-specific responses such as interferon. The effects of antibody and complement include the following. So, that's that. Now we're going to discuss the complement system, which I have summarized here. And the complement is on page 169. 169. Right here. The immune response, humoral versus cell-mediated immunity. Okay, the complement system is an array of approximately 19 plasma proteins and at least 9 membrane proteins that can act in a non-specific as well as specific way in host defense. One of the primary functions of the complement system is to assist the effects of antibody in the activation or destruction of pathogens. So it assists the antibody. Complement proteins in the plasma or other body fluids are normally in an inactive state. The proteins are activated by the presence of antibody on a cell surface, immune complexes, a surface component of microbial cell, blah blah. Here is a schematic of this complicated system here. Okay. Page 5. Okay. Well, that's... So we have C1. Okay. C1 protein is actually composed of 3 different proteins. Acts as an antibody recognition factor. That is, it recognizes binding sites on the antibody. After binding to antibody, C1 undergoes a conformational change and becomes a proteolytic enzyme whose substrate is C1. The substrates activated by C1 are C4 and C2. Cleavage of C4 and C2 produce peptide fragments C4A 2A, this convertase, and C4B associates blah blah, is enzymatically activated and called C3 convertase. Right there. The substrate for C3 is C3, which is cleaved into C3A and C3B. C3A acts as an anaphylatoxin, that is, it promotes C3A acts as an anaphylatoxin. It promotes a mast cell release of histamine, which affects vascular permeability and smooth muscle concentration. Produces an inflammatory response that can aid in the localization and destruction of infection agents by, remember, macrophages are activated by inflammation. Thousands of molecules are cleaved into C3A and C3B. Fragments can bind to cell membrane and interact components in the alternative pathway. There's an alternative pathway even. Blah, 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 blah. You see how complex this is. Okay. Ultimately, though, we get C5B. Okay. And gets attached to membrane. The final phase of the classic pathway involves formation of a multi-component complex. Okay. Binding of C5B to cell membrane begins the binding and section of complement to C6, C7, C8, C9 complex. And it affects this ultimately affects the permeability of cell membrane. This affects the flow of water into the cell, thereby causing lysis of the cell. It's called the membrane attack complex. Right there. And here's sort of a schematic of the cells here. Inserts into membrane. Okay. And then you've got an alternative pathway resulting in the same destruction. And so the membrane attack complex, the utilization of complement components in the formation of the membrane attack complex is called the complement fixation. It is an important clinical, and the complement fixation test, the reason I'm bringing this up, the complement fixation test is an important test, which, as I told you in the previous video, the book says that for coronavirus, the complement fixation test is the most reliable and the one that should be used. Now let's go to testing, page 563. 563. Now it took me, okay, complement fixation. It took me, I had to read this, I'm telling you, must have been 100 times, and finally I got it. Okay, and I've got it summarized here. Okay, the complement fixation test is where this is a test here, is where you add antigen, which is a virus, plus the test serum, which is, you know, your blood or whatever, plus complement, okay, plus sheep red blood cells. Okay, now normally if we take sheep red blood cells and antibody for that plus complement, you get a complement. Remember, lyse is a cell, and the sheep red blood cells, being a red blood cell, it causes a coloration, a red color, which is because a lot of times, like over here, I'll say, is because of viruses' small size, viruses generally do not give readily visible reactions with their homologous antibodies. Viruses with known effects in animals and/or in tissue culture are mixed with serum to determine the neutralization effect of the serum. That's for that test. Okay. So you get a pink coloration. Now on this test, it was really ingenious. They put the virus, okay, plus the test serum, plus complement, okay, and they let that sit. And then they add the sheep red blood cell. If antibodies are present, specific for the virus antigen, then complement is fixed to the antigen antibody. Okay, so you've got coronavirus and your test serum, and they add complement to that. And if you have antibodies in your serum, then the complement is fixed to the antigen antibody. So in a positive test, the complement is fixed or tied up and it's not available so that it's tied up, and the complement is not available to react with the sheep red blood cells. Therefore, there's no lysis of the sheep red blood cells, no color change. That means you have antibodies. In a negative test, where antibody is not specific, in a negative test where your antigen is not, in other words, your serum does not have antibodies, it doesn't combine, because remember, the antibody combines with the antigen and gets the complement cessation going. So it's not tied up, and when you add the sheep red blood cells, the complement instead reacts with that and it produces a red color, meaning you're negative for that. Okay. And I wanted to also say that in, let's say, the antibody test, which is a different test, over here. Okay, we've got, see, antibody titer test. The level of antibodies can be determined by using a constant amount of virus in following concentration of serum. See, a positive test in a single sample of serum is not a diagnostic value of an acute recent infection. Neutralizing antibodies can persist for years, remember in this chart? And their mere presence may indicate a past infection in a given individual. Thus, neutralization tests are useful in serologic epidemiology, where one is interested in knowing which viral agents have infected a given population in the past. Although simple in principle, neutralization tests are expensive in time and in materials and may be difficult to interpret, chiefly because of the variability of titration endpoints. See, so in certain instances, undiluted serum may be tested against falling concentrations of the virus in order to establish a diagnosis. One must be able to show a significant rise in antibody titer during the course of the infection. So you have to do, it's not just a one-time test, you have to keep sort of repeating it to see if you have an increase here, kind of quantitative, to see if you have that even. And like I said, over here, complement fixation test is a more sensitive index of human coronavirus infections than is virus isolation with a tissue culture or organ systems presently in use. Confirmation of infection by significant increases in antibody titer is more frequently successful using the complement fixation test than the neutralization test. These coronaviruses do not appear to be an important cause of acute respiratory illness in the pediatric groups. And I'll end with what I said before. The human coronavirus is extremely fastidious in its growth requirements, making routine testing difficult. So I'm basically questioning these little drive-by testing places, because even with a complement test, you leave this. So here, let's put this on the chart again and look at it. You have to, for instance, test procedure using heating serum, 30 minutes. The order of the test procedure is as follows. The test antigen and inactive test serum are mixed. A measured amount of complement is added. Complement source is fresh guinea pig serum, which is available commercially. After the mixture of the test complement has been, the indicator system is added to the test system tube. It's bound or not bound. The test not only is qualitative as described, but also can be quantitative. The complement fixation test is especially useful when the test antigen and antibody combination alone does not give a visible reaction. Lysis of the sheep red blood cells serves as a visible detection reaction. The diagnosis of many viral and fungal diseases and of immunological disorders is made or confirmed by this means. Well, what I was going to say is, over here, it was back in this book, you have to let this test sit. When you mix these reagents, I think they have to sit, let's see, yeah. No, I was on this page. Yeah, see, neutralization test, and then we have the complement test. So, well, anyways, they take, has to sit. It's not just a 2-minute test. So I think that's about it and what I wanted to say on the immune system. Basically, you've got an extremely complex system that's fully efficient and capable of fighting this cold virus. And I would not recommend taking a vaccine if God knows what they've even put in there. Thank you for watching, and please subscribe.